Thanks, Brett. Thank you, Brad. It's a pleasure to be here. And I am hoping in this talk to obviously convey the bottom line first, which is we desperately want and need more effective medications and interventional therapeutic treatments that really modify the disease and the diseases that we're dealing with. And we don't have those effective treatments at this point. I don't think that's a mystery for anybody in the room. So that's the bad news. The good news, in this talk, I want to talk about what the challenges are, where the complexities are, what are the barriers to that. You know, why does it seem to move slowly? Why don't we, year after year, why do we not have more in the way of promising therapies? But then I also want to convey why there's reason to be optimistic, which is really all of the work that's being done by the many people who are trying to address the complexities in the areas that need to be addressed to get to that point where we actually do have effective treatments. So the three main components of clinical therapeutic research studies that are really critical, we've already heard about them. I think I'm building on what our prior speakers have already spoken about. Number one is our biomarkers. Not just of molecular pathology, and by molecular pathology we mean those proteins, the tau, the TDP43, other proteins, but also biomarkers that are most relevant to the process of the diseases. And that's one of the areas that is challenging because we don't really have the final answers in terms of what the disease processes are, okay, and how those proteins relate to the disease processes. So a lot kind of builds on the research about those proteins and what roles they may play, but there also might be other complexities above and beyond the proteins, and we might need markers of those other complexities as well. The second major point is really that for these trials we need endpoints, which an endpoint is basically what you're measuring at time point A and time point B, and you're measuring the effect of your intervention. So what are you going to use to determine whether or not the treatment, in many cases the drug, is actually doing what we want it to do? So I'll talk a little bit about what studies have how they've typically approached this process in the past and in the current studies, but also kind of what some newer avenues might be to pursue. And then most importantly, we need the actual drugs that are going to kind of intervene on the disease pathways and slow them down or stop them, okay? So biomarkers, it's just a fancy term for tools that we use to make a diagnosis and then also track the progression of a disease. And so this is not an exhaustive list. This is an example of some things that we might consider biomarkers. When talking, there are multiple different types of MRI, some of which measure the structure of the brain, others measure kind of how the brain is functioning. PET, positron emission tomography, another biomarker, and there are different types of PET. And we've heard about PET that measures glucose metabolism, which is kind of a measure of how active brain cells are in a region of the brain. But then there are also these PET scans that do tell us whether or not a person might have an accumulation of a misfolded protein, like amyloid or tau. We have biofluids, so looking at cerebrospinal fluid and looking at the proteins or the other molecules in spinal fluid, and then also plasma, which is just part of blood. I mention this because I want to keep a global view. I try to keep my eyes on what's happening in other neurodegenerative areas, because there are common denominators across the whole spectrum, whether you're dealing with ALS or Parkinson's or FTD or Alzheimer's disease or or others. And so in a lot of these other areas, we are, like Alzheimer's, for example, there are promising efforts when it comes to blood tests that are not yet really available to be used, but there's an indication that they might be not too far off. And so it would be nice not to have spinal taps every time we want a biofluid marker, and I believe that this similar type of evolution is going to happen for FTD as well. So there is an important distinction when we're talking about biomarkers, because some of them, like MRI and FDG PET, they're really markers of just degeneration that's going to be nonspecific. It's not going to tell you what the disease is that's actually causing the atrophy or the loss of tissue. But it still is important for us to know what the topography is, what parts of the brain are affected. And then the markers of molecular pathology, like amyloid or tau, that are going to be more specific to the diseases that we want to intervene on and treat. So this is kind of a slide that really just is another way of kind of summarizing Dr. Dickerson's slide, where he was covering the three different kind of tiers of a diagnosis, kind of the second tier being a syndromic diagnosis, which is really defined by the symptoms and the neurological exam, versus the actual neuropathological diagnosis, which is what we see under the microscope, and the different kind of tools that we use. So there is a question, why do we have these associations at all? Why, as Megan said, that when we see a non-fluent primary progressive aphasia, our minds go to, well, this is likely to be tau. So it is because there's not a one-to-one correspondence, but there's kind of a predilection for certain pathologies for involvement of these large-scale brain networks that are actually responsible for generating the symptoms, because our brains are organized in a way anatomically that certain networks have different functions. So there is an association, and that's an unanswered question as to why. So that's one of the important questions about disease mechanism, is why are certain networks vulnerable to certain pathologies and resistant to others? Why do we have pathologies that actually might target more than one network, like Alzheimer's, which might cause aphasia, it might cause memory loss, but the same thing holds for these FTLD pathologies. Corticobasal degeneration might cause primarily a motor syndrome, but it also might cause a behavioral variant of frontotemporal dementia. And that's why it's super important not to really conflate the pathologies and the syndromes, because the way that we think about it is that the syndrome might arise due to any one of a number of different pathologies. So I think that point there is important to put in the context of therapeutic research studies, because if we don't have biomarkers for the molecular pathologies, we're not going to know what a person has going on under the microscope just by seeing what their clinical presentation is. We're not going to be able to distinguish between the behavioral variant frontotemporal dementia patient who has corticobasal degeneration pathology from PIC pathology, from TDP pathology. So there are a lot of different possibilities there, and if we want to develop disease-modifying treatments, then we want to know, most often we're going to be using drugs that have something to do with those proteins, or what's thought to be the mechanisms of why those proteins accumulate in the brain. So we need to, as a first pass, we need to select patients appropriately for studies. And this, again, using the Alzheimer's analogy, it wasn't until we had kind of spinal fluid and amyloid PET where we actually were able to say that, yes, people enrolling in these Alzheimer's studies have a high level of amyloid plaques in their brain, and as we know just from looking at kind of clinical pathological studies, about 15 or 20% of people who looked like they had Alzheimer's disease on the basis of their progressive memory loss and maybe even their pattern of atrophy on an MRI scan did not turn out to have high levels of amyloid, and they were enrolled in studies that actually involved kind of amyloid-based drugs, right? So we want to avoid that, and in order to avoid that, we need the biomarkers. So this is just a slide to show these kind of different protein subtypes and the fact that they look different, their different appearance under the microscope. We don't have a way, we don't biopsy the brain usually to determine these. So usually we're not going to have this diagnosis until we're able to look at the brain under a microscope in an autopsy. Okay. So endpoints, I think we've talked about already. Brad talked about it, and Bonnie talked about it a little bit as well, and so did Megan, in that a lot of our efforts are geared towards developing better endpoints that are more specific to frontotemporal dementia. We want, and endpoints might be either kind of endpoints that are looking at symptoms, or they might be endpoints that are looking at, they might be biomarker based. If we have a biomarker that we know tracks well with symptoms or has something to do mechanistically with symptoms, okay? So traditionally what we've done in studies is to kind of use standardized cognitive and functional batteries. So these might be some neuropsychological tests of function, as Bonnie was talking about. It might be kind of what we get from interviewing our patients and their caregivers and examining them. And primary endpoints in studies are typically meant to really capture both a cognitive element and then also just a functional element in terms of how a person's doing in their usual activities. And that's what the Food and Drug Administration wants to approve a therapeutic, is evidence of effectiveness in both of those areas, okay? But there's a problem in that when you're designing a trial and you're using a single, you have a single metric as your primary endpoint, you're going to be enrolling a lot of people that may have different kind of constellations of symptoms. And the endpoints might not capture the symptoms that are really relevant to the people, most relevant to the people who are entering in the study, or it so often occurs, people are excluded from participating because they don't actually have the symptoms in question. So I guess one classic example, again, going back to Alzheimer's, is that people need to have memory loss, really. So we can't enroll our patients who have kind of behavioral presentations of Alzheimer's or aphasic presentations of Alzheimer's, because they're not going to meet the criteria for the study that's designed to assess a memory endpoint, okay? So the Progressive Aphasia Severity Scale is an example of something that our group has done that is really, it's a development of a tool that's much more specific to kind of our FTD population, and can be used in clinical trials where we're enrolling people with PPA, and able to kind of track that in a much more meaningful way than kind of the Alzheimer's metrics. I think the other point that's very exciting is something that Bonnie touched upon, which is the ability to really take a more individualized approach, and that it takes it a whole step further, where you might be able to talk to the patient and the caregiver about what symptoms are most interfering in day-to-day life, and then really see whether your intervention is actually having an impact there. We were talking, Brad and I were talking recently about a case that was presented at a conference of a patient where the primary problem was the patient wanted to watch television, but was just having trouble operating the remote and was kind of just banging the remote on the table, and I think his wife wasn't, was interrupted from what she was doing so many times throughout the day because of this basic problem, this frustration of just inability to use the remote and the acting out and the frustration. And that's not something that might be captured in a single point of, one isolated psychometric measure that may not even be the primary endpoint in a therapeutic trial, right? So that's what matters the most to this family. But the studies are not designed to capture that in the least, and it would be useful to know if we were giving a treatment that and it provided a benefit for that symptom, that would be something that's useful to capture. So I think some of that variability, by taking a more individualized approach with some of the technologies, that's where things are headed. Those are the things that are being discussed when it comes to designing new studies. But I think more than anything, if we can understand the disease mechanisms better, then we will know which biomarkers are the most relevant, such that they can be used as the endpoints. And then we can shift it to even allow for treatment before people have symptoms, right? So that's something that we've taught, people might be aware of, that there are studies for Alzheimer's disease that are preclinical, where people get amyloid PET scans and then are receiving treatment and they're followed. I think that those studies are still relying on assessments of memory and thinking through time, because the amyloid PET isn't reliable enough. We don't know that simply lowering the amyloid on a PET scan is actually going to translate into a clinical benefit. Okay? So that's just an example of how the link between the actual proteins and the mechanisms of the disease is a super important link that needs to be elucidated. So this is a brief overview of the framework for kind of how new drugs are developed. And first we start off with kind of the bench research that's done, for example, with induced pluripotential stem cells and other types of models of diseases. And then if we can identify compounds that might be effective for the mechanisms of a disease, then we might try to generate models in animals of the diseases. And then if the drugs are doing what we want in the animals, then we would progress to studies in humans, those are the phase 1, phase 2, and the phase 3. And the phase 1 studies are really largely to look at toxicities and side effects, but also what we call the pharmacokinetics and pharmacodynamics, what is appropriate dose to use, does the drug get where we want it to go, how long does it stay in the system. And those studies are going to really inform in some cases there may be some components that might look at effectiveness, but largely effectiveness and side effects are more thoroughly mapped out in phase 2 and phase 3 studies. Usually it's going to be those studies it's always going to be those studies that the FDA is going to look at. And for FTD, because it's a rare disease, many times phase 2 studies are designed in such a way as to potentially provide the information that the FDA might use. So they might be a phase 2/3, so that you don't have to kind of enroll 100 people with a rare disease and then plan to enroll 1,000, and then you're not going to have your phase 3 study done for another kind of 10 years probably, right? So I think there is an awareness that that we want to be efficient in this process, but we also need to be diligent and not cause harm. And that's why we do need we need well-designed studies with a large enough sample. So on the last part, I'm just going to kind of briefly talk about some of the complexity, okay? And I'm not going to get too much into the weeds here, but the basic ideas are that many times, even when we're talking about the most simple scenarios of there being a single gene mutation that is passed in a family, there still are questions about how that gene mutation translates into a disease. And many times we're talking about loss of function of a protein that is important. Sometimes we're talking about what we call a gain of function, where an unhealthy protein might actually be toxic and tell a cell that it should die. And then there's also a concept that these unhealthy toxic proteins might spread. And one of the important advances in recent years was, in some of the basic research, was a demonstration that tau, unhealthy tau, appears to spread from cell to cell. And then downstream you see unhealthy tau. So this is a lot of detail here of just meant to kind of cover where some of the actual studied compounds are from recent past and some of the ongoing studies. I'm just going to highlight the fact that these anti-tau antibodies are actively being studied in phase 2 trials right now. The populations are largely PSP and then also Alzheimer's disease at this point. And the reason why PSP is selected is because it's a syndrome that very highly predicts that tau is the actual protein. There is a study of oxytocin, which is a hormone that is being studied in a phase 2 trial that's kind of being planned and in the building phases right now for symptoms of behavioral variant FTD. So the future, and this is, I know it's too much detail on the slide, but the big point is just that you have some cases that are due to kind of rare gene mutations that really cause a disease. And then you've got sporadic cases, which would be this area where we can't identify a single gene mutation, but likely there's a complex genetics and then complex genetic and environmental interactions that actually mediate the process of the disease. And what we're talking about with precision medicine is the intuition that we might need more specific mechanisms if we're actually dealing with different kind of disease mechanisms as being relatively more or less important for individual people, okay? So that's where there might even be a fourth level beyond the neuropathological diagnosis. It's the level of, you know, this particular case of PIC disease is related to this complex set of genes and environmental interactions where other cases might be due to others, and we might actually, in developing therapeutics, have to target the specific mechanisms applicable in different scenarios. This is something that seems a lot more intuitive to, I think, cancer doctors and researchers, you know, where it's just kind of accepted that there are so many mechanisms that are involved in cell proliferation. I think the involvement of these misfolded pathological proteins has actually been tempting for us to kind of view things differently in neurodegeneration, but my personal opinion is that the way that things are evolving is that there's this comparable level of complexity when we're actually talking about degeneration. And so when you actually look at single nucleotide polymorphisms that are associated with neurodegenerative diseases, you find that their relevance, the gene relevance, spans a huge broad range of functions from within the cell to kind of the local tissue environment to the body, which might be hormones, it might be inflammation, it might be vascular factors, and then other factors that really have to do with how the brain develops and goes through the aging process. And the point is that some different mechanisms might be selectively more or less important for individual people. So So that was my attempt to really capture kind of the complexity. And I think why should we be optimistic? I think, well, these efforts are all going on in parallel. So our group is largely working on the biomarker component and the endpoints component, but there are a lot of kind of molecular neurobiologists who are working out the disease mechanisms on a much more basic level, and that work is going on while we're doing our thing, and we put things together to actually achieve our goals. And we've come such a long way just in the last 10 years. We've identified TDP-43, wasn't known, 10 years ago. We've identified the C9orf72 gene mutation. We have learned a lot about the diseases over a short period of time, even though we don't have all of the answers. And so we're going to forge ahead, and hopefully we will be successful, and I'll be able to stand in front of you with good news in the way of more effective treatments in the near future. Thank you.
Thank you so much, Scott.